There we go. Hey, good afternoon, everybody. Hope you're having a great day so far. We are going to go ahead and pick up with the second half of the digestive system today, leading us to next week, diving into what do we do with all those nutrients? How does our body use them? Convert them to ATP and convert them from one type of macromolecule to another, depending on where there are shortages and surpluses, et cetera. So, just a word to the wise as we prepare for exam number 3. I would definitely encourage and strongly recommend don't miss next Tuesday and Thursday's lectures because the metabolism part is a little bit more involved. Okay. So I just like to share that up front. Last time, as you know, we introduced the digestive system. We learned that the digestive system has a variety of names: gastrointestinal tract, the gut, the digestive system, the alimentary canal. All of those are technically correct for the same system. We talked about the major organs and the pathway of the alimentary canal. We talked about some accessory organs that assist the digestive system. And the ultimate goal, of course, of the digestive system, the manipulation and digestion, both physical and chemical, of food down into the most basic molecules, the smallest version of our macromolecules, carbohydrates, proteins, and lipids, called monomers. And it's at that monomer level that they can pass through the walls of the small intestine, and those valuable nutrients can be absorbed into the bloodstream and then taken to the liver for processing via the hepatic portal circulatory system that we introduced. And then we certainly also talked about the value and the role of enzymes, and we even introduced the first enzyme, amylase, as starting chemical digestion already in the oral cavity. So that was an awful lot that we covered on Tuesday. We're going to go ahead and pick up from where we left off. If you recall, at the end of the lecture on Tuesday, we started working our way inferiorly through the components of the digestive tract, oral cavity. We did the pharynx, we did the esophagus, and then we even got through the sphincters, which again are just a fancier, different name for valves that the digestive tract uses. And that took us to the stomach. And that, of course, is where we'll pick up today. All right. So let's go ahead then and continue with the stomach. All right. So to no one's surprise, and long before you signed up for Bio 211, all of you realized that the stomach is a temporary holding facility, a storage unit for food in its pathway through the alimentary canal. So that's important, and that'll be a really easy one for you guys to know for the exam. However, what maybe is new to you is that much like starches and amylase, the enzyme starting to act on carbohydrates in the oral cavity, it's within the stomach where the first enzymes are introduced to start the chemical digestion of proteins occurs in the stomach. Okay, so that's a new layer. We haven't had any chemical digestion yet other than just of carbohydrates. Today we'll talk about and share the enzymes and where along the pathway lipids and proteins begin chemical digestion. And as we just shared, protein digestion begins, chemical digestion of proteins begins in the stomach. And we're going to talk about those enzymes and how that all occurs in the coming slides. I also mentioned in the last lecture that when we physically digest our food in the oral cavity, that, of course, then is mixed and converted with saliva from our salivary glands, those accessory organs, and becomes somewhat liquefied, paste-like, and is then termed a bolus. And from there, that bolus of food, that paste-like mix, can safely progress throughout the rest of the alimentary canal along the way. That goes all the way until and through the stomach. And then I mentioned previously, but I'll mention it again, that when the stomach gets done further physically digesting and chemically digesting our food products, the term changes from that of a bolus to that of chyme. So chyme is the exact same product, if you will, that entered the stomach, but when it exits the stomach into the small intestine, it changes terms from bolus to chyme. And really the only difference is the chyme is much more liquefied. Okay. It's almost the consistency of baby food at that point. And that serves an important point or value, because as I've mentioned previously, and we're going to talk about at length today, in the small intestine is where the nutrients, these monomers that are chemically and physically extracted from food, that all occurs in the small intestine. That's where absorption of nutrients occurs, where nutrients from our food passes through the wall of the small intestine into the capillaries that takes it to the liver. Okay, so the more liquefied that product is, our food is, that was first food, then became a bolus, then becomes chyme leaving the stomach. The more liquefied it is, the more exposed it is, and the easier it is for the small intestine to grab a hold of those monomers, those valuable nutrients. And the digestive system, just like other body systems in the human body, strives to be, intends to be as efficient and as effective as possible. Okay, so any nutrients, any monomers that pass through the small intestine without being absorbed are wasted value. So we're going to talk about several reasons and methods that the small intestine uses to enhance and improve its efficiency, its ability to capture as much nutrient value, absorb as much content as possible, and consequently excrete or eliminate as few valuable components as possible. The first of which is just that consistency of the mixed food, the chyme at this point. Being more liquid allows better absorption. Okay, we all knew in this class, before we even got to the digestive system, that our stomach can expand. Okay, and that's really because of the presence of rugae, and we talked about rugae in lab on Tuesday. The inner linings, these actual folds on the innermost lining of the stomach itself. Okay, and they allow for the stretchability of the stomach, that when we ingest a large meal, the stomach then can stretch to accommodate those volumes. And then as the stomach begins to empty itself back out, the stomach will contract back and get smaller again. Okay, so we talked about rugae. That should be pretty known to you at this point. All right, we also talked in lab on Tuesday about the 4 regions of the stomach. So this is just a recap. Of course, we had the cardiac region, that is the physical meeting place of the esophagus coming from above. The esophagus, of course, terminates at the cardiac sphincter, also called the lower esophageal sphincter. But that's where the cardiac region of the stomach is. It's a very small region of the stomach by volume compared to the others, but named appropriately for the cardiac sphincter that is embedded within it. Okay, and then we had the fundus, shaded here on this slide in a light purple color, which is the most superior top portion of the stomach itself, rather small also. And then, like I told you on Tuesday, the largest portion or region of the stomach by far is called the body of the stomach, and that's shaded here in pink. And then if we are following the passageway of bolus, to chyme and working its way down the alimentary canal, coming from the esophagus through the stomach. If we're following that pathway, the last of the four regions that bolus would travel would be the pylorus, that exit portion or region of the stomach, okay? And then from the stomach, that chyme is going to get ejected out into the small intestine. And there are three regions of the small intestine that we talked about in lab, the first of which is called the duodenum. And at the junction point between the pyloric region of the stomach and the duodenum, the first segment of the small intestine is the pyloric sphincter. So again, the anatomists kind of did us a favor here and named the sphincters, those valves, those intake ports and outlet ports of the stomach, after the region of the stomach that they emerge from. Okay, so the cardiac region is where the cardiac sphincter is. The pylorus region is where the pyloric sphincter is. All right, so let's talk about a couple of unique isms of the stomach compared to the rest of the alimentary canal. Okay, we talked last time here in lecture about the entire alimentary canal and the four tunics, the four layers that make up the tube, if you will, of smooth muscle with a lumen in the middle that is the alimentary canal. Okay, there are two significant differences with the stomach compared to all of the other organs of the alimentary canal, like the small intestine and the esophagus, etc. Okay, the first one is specifically looking at that muscularis externa layer. Okay, and if you recall to lecture on Tuesday, there were two layers of the muscularis externa that we talked about. There was a circular layer, which was the deepest of the two. And in that circular layer, the muscle fibers ran in a circular pattern around the lumen, the hollow opening in the middle. Okay, and then over the top of that, a little more superior or superficial to that, there was a longitudinal layer of smooth muscle that the fibers ran right over the top of it. And I told you that the reason that that's beneficial for having those two different layers of muscle fibers running in different directions was it increased the contractile force, the ability and the strength of contractions by having those contractions occurring in two different directions simultaneously. And that, of course, helps with peristalsis, the movement of bolus or chyme. Okay, so in the stomach, we actually have the first difference or nuance of the stomach is that there's a third layer to the muscularis externa. And the stomach is the only place where this third layer is found. And this is yet a third layer that is the deepest layer, and it's called the oblique layer. And in this case, the muscle fibers run on an angle. Okay, so we had the circular layer of muscle tissue. We had the longitudinal layer running lengthwise for all of the alimentary canal. But when we get to the stomach, we also have an oblique layer. And if we go back here, we can actually see these layers, the longitudinal here on the top, the circular layer laying here in the middle, and then this new layer introduced, which is now the deepest layer, the oblique, and you can see the muscle fibers running on an angle. And all of this is only to add yet additional contractile force, okay? This may not seem common sense-ish at all, but the stomach is the second most involved player when it comes to physical digestion of food. Of course, first is what we talked about at length last time, our oral cavity, where our teeth and our tongue and our salivary glands all come together and physically take food, a cheeseburger, and mash it and turn it down into a saliva-coated paste that's called bolus, right? But so that's pretty obvious. Everyone could guess that physical digestion would occur in the oral cavity. But not everyone would naturally think that physical digestion would also occur very aggressively in the stomach. And it's because of this third layer of the muscularis externa that really causes the muscles of the stomach to churn and mix and just smash and maul food to make it even more liquefied. In other words, turning the bolus that arrived into chyme, a much more liquid version for the reasons I shared with you to enhance absorption in the small intestine. But that physical digestion of the stomach is very important. Okay? So, and then along with that, like I mentioned previously, in the stomach is going to be where chemical digestion of proteins is also introduced, and we're going to talk about that next. So the first subtle change, third layer of muscle in the muscularis externa, called the oblique layer, and it's all about additional physical digestion to turn that bolus into chyme. All right, the second variation within the stomach versus the rest of the alimentary canal is the innermost lining. And if you remember back to Tuesday, the mucosa was the innermost lining of the alimentary canal as a whole. Okay, that still is going to remain true. However, within the stomach, the mucosa, the innermost lining, is different in the type of tissue that makes it up. Now, when we're talking about the inner lining of the stomach itself, okay, it's going to consist of, and that mucosal innermost layer is going to consist of, back to Bio 210, those simple columnar epithelial tissue. One layer of columnar-shaped epithelial tissue. And if you remember, what's really unique about simple columnar epithelial type cells and tissue is they contain goblet cells. Okay, and goblet cells secrete, they create, and then expel, secrete mucus. Okay, and so that mucus is important throughout the body in various systems for lubrication. But in this case, when we're talking about the lining of the stomach, it has a second benefit, the mucus secreted by those goblet cells. And that's that the mucus that is secreted in the inner lining of the stomach is alkaline based. All right. And that's really, really important. Remember, when we talk about pH, we have alkaline and we have acid, right? And we all know that our stomach produces hydrochloric acid. We're certainly going to talk about that in the coming slides in more depth, okay? But that hydrochloric acid that's made and released in our stomachs to aid with digestion and further physically and chemically digest our food, to break it down, is very aggressive. It's extremely corrosive. In fact, of all the acids in the human body, our hydrochloric acid in our stomach is the most acidic. It's the most violent. It's the most aggressive. If you remember, pH of 7 is completely neutral. As we move from 7 on the scale down towards 0, that acid becomes stronger. Okay? The pH, the acidicness of hydrochloric acid is somewhere around 2. So it is extremely acidic. It will eat practically everything. It has the ability to dissolve practically everything. Okay, so because of that, that's a double-edged sword, okay, or a blessing and a curse. It's very good in that hydrochloric acid can aggressively dissolve and decompose things that are very solid, things that have great density, like fiber or steak, very dense proteins. that don't easily dissolve otherwise. That acidic nature of hydrochloric acid is aggressive enough to cut through it and liquefy it. Okay, so that's the blessing of it. The curse is that aggressive nature and condition level of hydrochloric acid can do tremendous damage to the tissue that it comes in contact with. Okay, and the first point of contact is going to be the inner lining of the stomach itself. All right, so the stomach takes protective efforts by having these surface mucosa cells with goblet cells secreting mucus filled with alkaline, which is a base, to lay down a protective coat of alkaline around the inner lining of the stomach to try to some degree, neutralize the effects of hydrochloric acid. So the acid doesn't burn into the surface of the stomach, causing injury, trauma, or what we call ulcers. Okay? People who have stomach ulcers, they can come for a variety of reasons. But one of the reasons at times when people have stomach ulcers is because these surface mucosa cells aren't secreting enough alkaline-based mucus. And then that hydrochloric acid has free reign and starts burning, causing ulcerations, burns into the stomach itself. Okay? So that's what's unique about the entire inner lining of the stomach itself. Now let's take a look at what we call, and we see here in this slide, illustrations of a gastric pit and a gastric gland. And remember, I told you we talked more about this in lecture today during lab. We saw the histology slide of gastric pits of the stomach that I told you to make sure to remember for the lab quiz, okay? And if you remember, gastric pits are just hundreds of thousands of tiny, tiny little pinhole-like indentations in the inner lining of the stomach. Here we're looking at a lateral view of that, but that's what this is, just a tiny little one gastric pit. And the gastric pit, by definition, really is just this initial opening of the pore. And if we really go all the way down to the depths of it, to the very base, you can see the majority of it is considered technically a gastric gland. But at the end of the day, guys, these are all just very tiny indentations all along the inner lining of the stomach. They're gastric pits combined with gastric glands deeper down to them, okay? And from these cells that make up the gastric gland are going to come a variety of things, all right? So these next few slides that are going to relate to, of course, chemical and physical digestion, as well as some regulatory control of the stomach. All right, so what we're going to do now in the coming few slides is go through each of these varieties of cells as we explore deeper into the gastric gland. We talked first about the mucus cells and laying down alkaline-like mucus across the entire surface of the stomach to protect ulcers because of hydrochloric acid. All right, so what I'm going to do is bop back and forth so we can see the cells that we're talking about. The first one we're going to talk about are called mucus neck cells, okay? So going back here to the illustration of the gastric gland, as we dive deeper in past the surface cells that we've just talked about and into these mucus neck cells, this narrow bandwidth here of these type of cells, what these mucus neck cells do is they lay down as the name says, a mucus, but in this case, the mucus is actually acidic in nature. It's not alkaline based like the surface mucus cells are, okay? So the best I can tell you about that is we know that that's what the mucus neck cells do. We know that they produce and secrete a mucus that is acidic in nature, not alkaline. Okay. Beyond that, scientifically, we really don't have an explanation. And there are times with the human body that we, quite frankly, just don't know why physiologically that occurs. There really is no rational reason for these mucus neck cells to synthesize and then excrete a acidic-based mucus. But nonetheless, that's what occurs. So that's what I can report to you. Okay? Next, we'll talk about, as we move deeper into the gastric gland, we're going to talk about these parietal cells that lie on either side, right underneath the mucus neck cells. And this is where things really do get interesting. This is where hydrochloric acid is synthesized, created, and then secreted. Okay? So this is where the hydrochloric acid, that is that chemical and physical breakdown mechanism within the stomach, that very aggressive acid. As you can see, I said, that somewhere around 2, the range is somewhere between 1.5 and 3.5, but nonetheless, the number is irrelevant. It's extremely aggressive as an acid. It is going to denature protein. In other words, start to actively, physically digest and soften up proteins, like I said, okay? It's going to actively engage and break down fiber, plant cell walls, things where otherwise physical digestion wouldn't necessarily liquefy them. Hydrochloric acid will, okay? Along the way, we talked about in the oral cavity that in food and beverages that we ingest, sometimes there are bacteria or pathogens or something harmful that comes attached to our food. If something made it as far as the stomach, the extremely acidic nature of hydrochloric acid helps to kill off any bacteria or threats that might make it this far. Okay, so a variety of things that hydrochloric acid do. Okay, however, maybe one of the most important, to be sure, functions of hydrochloric acid is to activate pepsinogen and convert it to pepsin. Let me explain what that means. We're going to see on the next slide where pepsinogen comes from. Spoiler alert, it's deeper into the gastric gland. Okay? Pepsinogen is an enzyme that is secreted from the gastric glands into the stomach. And we know that enzymes are very much responsible for the chemical digestions of a very specific macronutrient, like a carbohydrate, a protein, or a lipid. All right, pepsinogen, again, is an enzyme secreted by the gastric glands into the stomach. That pepsinogen just hangs out in an inactive but ready to respond state, okay, until it meets up with hydrochloric acid, all right? And when hydrochloric acid is released from these parietal cells and interacts with the enzyme pepsinogen, which has been just hanging around waiting to act in the stomach, it's going to convert the enzyme pepsinogen to the enzyme pepsin, which is the enzyme that's going to chemically digest protein. Exactly how amylase chemically digested carbohydrates, starch, already in the oral cavity, okay? So remember I said, the initiation, the first efforts of chemical digestion of proteins, reducing them down to their monomers, which we know are amino acids, is because of the enzyme pepsin. But pepsin comes from pepsinogen only after connecting with hydrochloric acid. Okay, so a really important function. So 2 really important functions of hydrochloric acid: breaking down very difficult, dense food product, and then converting pepsinogen to pepsin, and then pepsin in turn, that enzyme will start to chemically digest proteins and turn them into amino acids. So again, when we get as far as the small intestine, we can absorb them into the bloodstream. All right. The second thing that the parietal cells do, okay, so secretion of hydrochloric acid, number 1. Number 2, secretion and creation of what is called, it's another enzyme called the intrinsic factor. And you may remember from the video that we saw in lab on Tuesday. He talked about the intrinsic factor and its relationship with vitamin B12. You can see this is underlined, so it's really important for you guys to understand this. The enzyme intrinsic factor has to be attached to vitamin B12 at the small intestine level in order for the vitamin B12 to be absorbed. In other words, for B12 to pass through the wall of the small intestine, through the wall of the capillaries and into the bloodstream, and then distributed to the whole body. Okay? If vitamin B12 comes through the stomach from our diet or from a multivitamin or from whatever, and does not mate up with the enzyme intrinsic factor, vitamin B12 will just continue to flow through the small intestine, through the colon, and be eliminated as waste. They have to be coupled up. Think almost back to the endocrine system when we had hormones that needed a very, very specific lock and key in order to activate them, right? That's the relationship between intrinsic factor and vitamin B12, okay? This is the only function that the stomach produces that is absolutely essential for our very survival, okay? Because vitamin B12 is an extremely vital vitamin to our existence, because vitamin B12 is the major vitamin responsible for erythropoiesis. Remember back when we talked about blood, and erythropoiesis is the creation of new red blood cells, erythrocytes? If we don't have an adequate supply of vitamin B12 extracted from our digestive system and making its way into the bloodstream, because there's not intrinsic factor, then we can't make new red blood cells. And if we can't make new red blood cells, we all know how vital they are for oxygen carrying. All of our organs would start to shut down very rapidly, and we'd be in big trouble. Okay, so that's why intrinsic factor, it's an enzyme released by the parietal cells of the gastric glands, and it is absolutely required to bond, couple, mate up with vitamin B12. Once that occurs, B12 can pass through the intestine wall and make its way through the body to facilitate further production of new red blood cells, erythropoiesis. All right, let's go next to the chief cells. Let's go back for a second. Again, we're just moving deeper into the gastric gland, as I told you, okay? Now we're below the parietal cells that create and release hydrochloric acid and intrinsic factor. Now we're down to these chief cells. We're near the very base of the gastric gland itself, all right? So the chief cells, whoops, went the wrong way. Sorry, guys. Chief cells. This is where pepsinogen comes from. Remember, I promised that we'd find out where does it come from. I told you it comes from the gastric glands. That pepsinogen, that enzyme that is inactive until meeting up with hydrochloric acid, and then it changes to pepsin, and pepsin is the protein enzyme that chemically digests protein. Okay, that's where the pepsinogen comes from in the first place, is within the chief cells, okay? And again, pepsinogen and the eventual mating of pepsinogen and hydrochloric acid to change pepsinogen to pepsin is what facilitates and allows the chemical digestion of proteins in the stomach. So very, very important, because we certainly want those proteins to be monomers. We want them to be amino acids by the time they get to the small intestine, so we can use them and not excrete them as waste. All right? And then the last one at the very base of the gastric gland are going to be these enteroendocrine cells. Enteroendocrine, just from their name, you should start to get a little bit of a guess, at least in form, of what they do. The enteroendocrine cells secrete the hormone gastrin. Okay, as soon as we saw the root word endocrine, from our knowledge, what we've already learned, we know the endocrine system is responsible for monitoring hormones, right? And production of hormones. We didn't talk about gastrin as a hormone when we talked about the endocrine system, because as a system-wide for endocrine, gastrin is a relatively minor player, okay? And plus, we cover it when we come here to the digestive system, okay? But know that these enteroendocrine cells, also at times called G cells, because of the hormone that they create and release, gastrin, this is where gastrin is produced and released, at the very base of the gastric glands, okay? And gastrin, we're going to talk about it more in the coming slides, how it does this. But gastrin is a local chemical messenger. And remember, we talked about local chemical messengers, meaning that it's a hormone that can send a signal within the same organ, okay? And it also circulates as a hormone like others. So it can take and send signals elsewhere in the body. But really, gastrin is, for the most part, a local chemical messenger, meaning it's a hormone that's going to act right there locally, right in that region, to cause something to happen or cause something to stop happening. We're about to find out. All right, so those are the gastric glands and what all of these cell types do as we descend into the glands. Okay, very important. Again, we saw some protective type effects as far as alkaline base and warding off the harmful effects of hydrochloric acid. We saw some hormones being released. We saw the hydrochloric acid and some enzymes being released that are responsible for both physical and chemical digestion. So a lot of different things going on from these gastric glands. All right. So the digestive processes, what are the things that occur beyond a temporary storage facility for food in the stomach? And you can see that this is underlined, so it's going to be important that you know these for exam number 3, okay? And we've talked about all of these already, so this is going to be a recap. And the stomach, again, beyond the temporary storage facility, secretes that intrinsic factor, that enzyme that's vital to life, coupling up with B12. We learned about the third muscular layer, the oblique layer of the muscularis externa, that allows for much more aggressive contractions, leading to more and significant additional physical digestion of food, really helping to convert bolus, a paste, to chyme, more of a liquid, which is important for absorption. The hydrochloric acid is going to start to break down proteins. Pepsin is going to start the chemical digestion of proteins in the stomach. We have water absorption that occurs within the stomach. Water, remember we talked about, is the only nutrient that's absorbed in 3 different locations of the digestive system: the stomach, the small intestine, and to a lesser extent, the large intestine or colon, okay? Everything else, nutrient-wise, is absorbed in the small intestine. Water can be absorbed in all 3 places. So water is absorbed from the stomach because water is, of course, our most valuable nutrient. Our body needs water more than it needs anything else. We are, as a body, a human body, almost 2/3 water in composition, okay? But beyond water, lipid-soluble alcohols and aspirin also can pass through the walls of the stomach into neighboring capillaries, all right? And then ultimately converts bolus to chyme, which then is ejected into the small intestine, all right? So a lot of different things that the stomach does, all things that we've talked about now in detail. It should be pretty easy for you when it comes test time. All right, so gastric juice secretion, all right? So what regulates how many gastric juices and the rate that gastric juices are secreted, okay, in the stomach? And like so many other systems that we've talked about through Bio 210 and this thus far through Bio 211, there are both neural mechanisms from the nervous system that either turn things on or turn things off or speed things up or slow them down. And then there are also hormones involved that turn things on or turn things off or speed things up or slow things down. Okay. And usually, as we've seen, most systems have contributions or regulatory mechanisms from both the nervous system and the endocrine system to helping to regulate things, ramp things up or slow them down. When we look at the nervous system and the impact on gastric juices, and we can see our gastric juices secreted a day, roughly a little more than 3 liters a day. Okay, and we all know what a liter of soda looks like as far as volume, right? 3 liters of soda per day produced by our stomach. That's an awful lot, right? So regulating how much so that there's not too much or enough so there's not too little is very, very important to a healthy digestive system and healthy absorption of nutrients. So from a nervous system impact, we'll start with the vagus nerve. Okay, this should take you back to Bio 210 when we talked about those 12 pairs of cranial nerves. The vagus nerve was one of those cranial nerves, one of those pairs of cranial nerves. And if you remember, that was one of the outliers. It was one that really didn't quite fit in with the others, in that most of the cranial nerves had local control of something like special senses related to our sense of smell or our hearing or our vision, right? Our optic nerve, for example, with our vision. Okay. The vagus nerve is sending input all the way down to the stomach. It's not, most of the cranial nerves regulate things around the head in the cranial region. But the vagus nerve was very different in that it reaches and sends signals and regulates things all the way down as far away as the stomach. So that's what makes it unique. Okay. If you remember the nervous system, there's parasympathetic and sympathetic, or are opposites of one another. Okay. The vagus nerve is part of the parasympathetic nervous system, and it stimulates and increases the secretion of gastric juices from a nervous system standpoint or control point. And then the opposite of that is the sympathetic nervous system then slows things down or decreases the secretion of gastric juices. Okay, so those are your nervous system inputs, parasympathetic and sympathetic, the vagus nerve being that parasympathetic that's going to increase the secretion, the release of gastric juices. From a hormonal standpoint, okay, here's gastrin. Again, remember I promised that we talk about gastrin and what that hormone does. And I said that it was a local chemical messenger. This is that local chemical messaging. The hormone gastrin, released by those enteroendocrine cells at the very base of the gastric glands, stimulates the secretion of hydrochloric acid by those parietal cells right above them in the gastric gland. Okay. And that's very important because, again, hydrochloric acid has to interact with pepsinogen to turn it to pepsin. Okay. And then the opposite hormonally is the hormone secretin. And we'll see where that comes from in a minute. But secretin is the hormone that works opposite of gastrin, and that's going to slow down or stop the production of gastric secretions from a hormonal standpoint. Okay, 2 different systems, turning things up, turning things down, just like we've talked about with so many other body systems. All right, then we have enterogastrones. Okay, so first of all, enterogastrones, we're still talking about the stomach, okay? But these are cells that are located actually in the duodenum. And you know from lab, the duodenum is the very first segment of the 3 portions of the small intestine emerging off of the stomach, the duodenum, only about 12 inches or less in length, okay? The duodenum has enteroendocrine cells built into the walls, and the duodenum, and specifically the enteroendocrine type cells, release two important circulating hormones that relate to gastric secretions, okay? So gastric secretion, the rate at which food passes out of the stomach into the small intestine, okay? So the first one's very important, and this is that hormone secretin that we just talked about, right? Secretin is going to slow down or stop gastric juices, hydrochloric acid, right? From a hormonal standpoint. Secretin comes from these duodenal enteroendocrine cells, okay, and is released when the chyme that's coming from the stomach is too acidic in nature, okay? And you can imagine any chyme emerging through that pyloric sphincter into the duodenum, into the small intestine, all of that chyme is now coated with hydrochloric acid, just like the bolus was coated with amylase when it continued through the pharynx and the esophagus. All right, so the trouble we run into is if there's too much, if it's too high of an acid content, we can do damage to the walls of the small intestine and or colon. All right, so when that chyme is coming out at a dangerously high level of acid, secretin is going to be released from the duodenal cells and sending a signal to target the parietal cells of the gastric pits again to slow down or stop the production of hydrochloric acid, to bring the acid levels down so the chyme emerging out of the stomach into the small and large intestine aren't quite as acidic, and there isn't the risk of damage to the walls, okay? So that's related to the pH of the chyme and secretin's involvement. Then we have cholecytoskinin, CCK, another hormone that is stimulated by if the chyme emerging into the duodenum from the stomach is too fatty. There's too many lipids in it, okay? And when chyme has too much fat content in it, that is a sign that it should have spent more time in the stomach, being further digested. So in that case, the duodenal enteroendocrine cells will release CCK that are going to target the muscularis externa layer of the stomach itself as a local chemical messenger and decrease stomach contractions, slow them down so that the food can, the bolus at that point technically, can spend more time in there. It's going to slow down the emptying of the stomach itself. So there's more time for further digestion, reducing the ratio of fat in the chyme. All right, so that's what these hormones do and where they emerge. So then we have regulation of gastric emptying, okay? The pace at which the stomach empties itself. All right, so very much related to gastric juices, by the way, okay? But gastric emptying is really controlled by that pyloric sphincter for the most part, okay? That valve, that sphincter that connects the pylorus, the most inferior of the four regions of the stomach, with the duodenum, the first part of the small intestine, okay? It's that sphincter that monitors what's going on already in the small intestine, and only when it's ready will open and allow more chyme to exit the stomach and make its way in to the duodenum, okay? So it doesn't overfill, because if the small intestine was overfilled and too much chyme was just free flowing through it, there wouldn't be enough time for proper absorption, and we would miss the opportunity to absorb nutrients. And the small intestine and the digestive system as a whole would become inefficient or less efficient. In general, our stomach empties, as you can see, approximately every 4 hours. However, there are certain circumstances that might speed that up. If we ingest a lot of lipids or carbohydrates, things that are very easily digested, they tend to pass through the stomach much faster. Okay, if you are on a liquid-only diet and you're only eating chicken broth, those liquids that would be your quote-unquote food don't need nearly as much digestive time. They're going to pass through the stomach much more rapidly than 4 hours. Okay, and then also affecting the rate of the stomach emptying and the time that it empties itself is in relation to how much food we've taken in. If we eat a tremendously large dinner this evening, if you remember, I used this as an analogy before when we talked about the gut brain, our enteric and intrinsic nervous system, that nervous system that allows the digestive system from one part of it to communicate directly with the other. If the stomach ingests a lot of food at one meal, it is going to use that intrinsic nervous system and send a signal to the colon, to the large intestine, to speed up contractions and enhance peristalsis to start clearing itself out. So we're making room for the food that's now in the stomach. Okay, so a large meal tends to speed things up because that gut brain is going to send signals to speed things up because a lot of food has been introduced to the area. All right, first of your 2 quiz questions today. So beyond storage and additional physical digestion of food, what does the stomach do? Oh, let's continue. All right, so now we're on to the small intestine proper, even though we've been talking about things occurring in the duodenum because they're related to the stomach. We're not going to spend a lot of time on this slide. We talked about this at length over in lab, right? The 3 segments of the small intestine, you definitely need to know them. And in the order of which they occur in the alimentary pathway, the duodenum becomes the jejunum, becomes the ileum, and then the ileum turns into the large intestine at the cecum section of the large intestine. Again, the anatomists did us a favor in one way, and there's an oddity in the other, and that the ileum section of the small intestine physically comes into contact and becomes the cecum section of the large intestine. So the gate that's going to allow chyme and waste to proceed through it is called the ileocecal valve. That makes sense. Ileum and cecum, right? So ileocecal. But for whatever reason, and I have no earthly explanation why this would be, but the ileocecal, this is called a valve for the first time when talking about the digestive system. So far, we've had the upper esophageal sphincter, the lower esophageal sphincter, and or cardiac sphincter. We've had the pyloric sphincter, and now for some reason, they changed the terminology and call this one a valve, okay? But nonetheless, that's what it is. All right, so back to some additional accessory digestive organs. Remember, on Tuesday, we talked about the teeth, the tongues, and the tongue and the salivary glands being accessory digestive organs working toward the physical digestion and chemical digestion in the oral cavity. The liver, the gallbladder, and the pancreas are all accessory organs of the digestive system, and they contribute for the most part to chemical digestion, but to some degree to physical digestion as well. And we'll see why in a minute. Okay, so we'll talk about all of these individually, and you'll understand what's occurring here and how these accessory organs contribute to digestion. All right, so let's start with the liver. All right, first of all, the liver makes bile. Okay, our liver makes bile, and bile can be broken down. A component of bile are called bile salts, and bile salts emulsify lipids, fats that we take in our diet. Emulsify means to break down. This is physical digestion, okay? It's kind of like hydrochloric acid emulsifies protein like steak, okay? It kind of liquefies it. It doesn't chew it like our mouth, but it is a type of physical digestion because it's physically changing it from a solid or from a semi-solid to a liquid, okay? So the liver makes bile. A component of bile are bile salts, and bile salts physically digest fat, okay? So that's the example of physical digestion that happens from these accessory organs, the liver, the gallbladder, and or pancreas, okay? Now, the liver can deliver bile via ducts. Remember, ducts are just tubes that carry something from one organ to another, in this case, bile. The liver can deliver that bile that's produced in the liver through a duct straight into the duodenum, that first segment of the small intestine, via a duct. Okay? So that's first. All right, let's move on and talk about the gallbladder. The gallbladder, which we talked about in lab, is kind of avocado size and shape. It's kind of embedded in the posterior aspect of the liver. And it works hand in hand with the liver, like I talked about in lab the other day, on Tuesday. The gallbladder is nothing more than a storage shed for extra bile that the liver produces. The liver produces bile. It can deliver bile direct to the duodenum to start physically digesting fats. And at the same time, if it's producing more than enough or more than is required bile, it'll send that bile to the gallbladder to be a temporary storage location in case there's ever a shortage coming up. Okay? The gallbladder has ducts also that can take that bile that's stored in the gallbladder and deliver it right to the duodenum for the physical digestion of fats. So bile can get to the duodenum from either the liver or the gallbladder. All right. And then the pancreas is a major player when it comes to chemical breakdown of carbohydrates, fats, and proteins, those macromolecules into monomers. For the first time, we have the pancreas, first of all, produces what's called pancreatic juices. Okay. The enzymes that are produced from the pancreas are called pancreatic juices. And those pancreatic juices have the enzymes to break down all three: carbohydrates, proteins, and lipids chemically. Okay. Previously, we've seen amylase breaks down carbohydrates and pepsin breaks down proteins. But we don't even have something until we get to the small intestine and introduce pancreatic juices that's even started to chemically break down fats, right? So pancreatic juices are extremely important to supply enzymes to chemically digest all three of the organic molecules into monomers. So that's why the pancreas is such an important player. And as we all know, from the endocrine unit, the pancreas also has a tremendous importance when it comes to endocrine function to monitor blood glucose levels, right? Insulin and glucagon, hormone regulation. So the pancreas is a really, really important organ. All right, so we've already seen this over in lab, but here are some illustrations that show the relationship of the liver up above, the gallbladder sitting back here, like I said, kind of an inferior, posterior aspect. We see the ducts here, the tubes that connect the liver to deliver bile all the way down into, this is the duodenum of the small intestine. Here's the duct coming right through the wall here, that bile making its way straight from the liver. The gallbladder, like I said, is a storage facility for extra bile. The gallbladder, if needed, can release that bile through its own duct down in and through and into the duodenum to, again, physically digest lipids. And then the pancreatic duct we talked about in lab also, that runs lengthwise down the length of the pancreas, where those pancreatic juices are produced all along the way and make their way down this pancreatic duct. And the pancreatic duct comes together with the bile duct from the gallbladder and liver right here in the wall of the duodenum. So pancreatic juices and bile from either the gallbladder and or the liver all make their way through the same little opening here in the wall of the duodenum. Okay? So that's the gallbladder recap of how it works with the ducts. Here's the pancreas. I just talked about that on the previous slide. Okay? Pancreatic duct carrying the juices to the duodenum. All right, so what is pancreatic juice besides all three of the enzymes needed for chemical breakdown of carbohydrates, fats, and proteins? Okay, mostly it's water. Like any other fluid in the human body, it's water-based. It's slightly base in nature or slightly alkaline in nature. Really, anything downstream from the stomach, anything that gets introduced to the system is going to tend to be alkaline based for obvious reasons. Again, the chyme that's emerging from the stomach is going to be coated with hydrochloric acid. It's going to be it's acidic. So anything that is going to be introduced beyond that is going to be alkaline in base to try to neutralize the harmful effects of the acidic nature of hydrochloric acid. Okay? Beyond the alkaline water type liquid, we have those enzymes that we talked about that are vital to chemical digestion. For proteins, those enzymes are proteases. For carbohydrates, it's our old friend amylase, yet again, the exact same amylase enzyme that's released from our salivary glands in the oral cavity that starts breaking down starches in the oral cavity. This is the same amylase, but this is a second dose of it released as a component of pancreatic juices, again in the small intestine. And then for lipids and fats, lipases are the enzyme. Remember that one for the test, that lipases are the enzymes as a component of pancreatic juices that chemically digest fats. All right. So our bile release and pancreatic release are controlled hormonally. Okay. There are also neural controls, like so many, that both will stimulate the release of bile and pancreatic juices, but for the most part, hormones drive these releases, okay? So we have CCK again, and we have secretin, are both hormones that are going to be local chemical messengers that are going to cause the release of bile from the liver or the gallbladder and pancreatic juices from the pancreas. All right, let's move to the small intestine proper, and this one is a recap. Again, both to lecture and lab. the three parts of the small intestine. Know for the lecture exam that, again, the majority, and I mentioned this so many times, I don't know how you possibly couldn't know this at this juncture, that the vast majority of absorption of nutrients, the extraction of valuable nutrients from the food that we started with, occurs in the small intestine. Okay? No surprise there at all, right? Also, most chemical digestion takes place in the small intestine, and that makes sense based on what we just learned about pancreatic juices, right? Enzymes for all three organic molecules being released into the duodenum. So naturally, the majority of chemical digestion would occur in the small intestine as well. All right, now, when we get to the small intestine as a whole, and we've talked about these subcomponents or additions, to the small intestine in lab. But the small intestine's job is absorption, that grabbing a hold of any nutrients passing by and pulling them through the intestinal wall into the capillaries so those nutrients can be spread throughout the body and used at the cellular level. So because the primary job of the small intestine is absorption of nutrients, increasing surface area of the small intestine is vitally important. And the small intestine increases its surface area, much like, if you remember back when we talked about the respiratory system, our alveoli, right? Trying to increase surface area for more efficient gas exchange. The small intestine increases its surface area for more efficient nutrient absorption, okay? And we talked about all three of the ways that the small intestine is altered or modified compared to the rest of the alimentary canal. What's different about it that increases surface area? First were the circular folds that we talked about in lab and we saw pictures of. Remember, they look kind of like the rugae, those ridged inner linings of the stomach. But in this case, the small intestine doesn't need these ridges to expand like the stomach does. It's just sheerly putting ripples into the inner lining, increases contact area. It increases surface area, okay? And then what you do need to remember for the lecture exam are things that we talked about in lab already. And those are those finger-like projections off the inner linings of the small intestine called villi, finger-like projections. And then off of the villi, we can see here in the inset in this blow-up image, we can see even further, almost microscopic, almost hair-like, finger-like projections off the villi themselves that are called microvilli. Okay, so you can imagine the small intestine if it was just a smooth, hollow tube like the esophagus, for example. The surface area would be far less than if you put all these villi and little microvilli popping off. We've increased the surface area significantly. And again, increased surface area contributes to improved performance, specifically the absorption of nutrients, the very job of the small intestine. And once again, comparable amount of surface area of our small intestine. If we lay them out flat, all three segments of our small intestine, they'd be roughly the size of a tennis court again, which, if you remember, is not ironically the exact same estimate of surface area of the alveoli of our lungs. So if you think about it, it's pretty amazing that within our lungs, within our thoracic cavity, we have essentially a tennis court rolled up in there that tightly. And then within our abdominal cavity, not even including the stomach or the large intestine, we have another tennis court rolled up in there. And it's pretty astounding if you think about it. All right, let's move to the large intestine and colon to wrap up today's lecture, guys. All right, so we talked about these parts. So I'm not going to spend as much time with you as I did with my morning lecture where I don't have them for lab. Again, the whole point of lecture and lab typically being different professors is you hear it twice from different people. I know that I've covered this at length in lab on Tuesday. But remember, the ileum, the last segment of the small intestine, articulates with the first segment of the large intestine, and that's right here, and it's called the cecum. And following the pathway of chyme or waste at this juncture, we go from the cecum through the segment called the ascending colon. We make a turn. We start horizontally across the abdominal wall, transversely across in the transverse colon. We make yet another turn, and now the waste product starts descending through the descending colon. And then right before the colon or large intestine terminates, there's a little J-shaped segment called the sigmoid colon. And then the colon, the large intestine, technically stops, and it becomes the rectum and eventually the anal canal for waste excretion, okay? You guys know these parts. You definitely need to know these parts for the lab quiz. Just if you know them, you're in great shape, okay? Off of the cecum, we mentioned in lab that we have this little finger-like projection called the appendix, which for years we thought was an obsolete, outdated vestigial organ. We now know that we were wrong. Microbes are produced here by the millions and then spread all throughout the large intestine, which we'll talk about in a minute. All right, so unique to the large intestine is from a physiologic standpoint. And this takes us back all the way back to day one of Bio 210. The very first thing in lecture that we talk about in Bio 210 is that in anatomy, form equals or meets function. Okay, form and function complement one another in the human body. It's called the principle of complementarity, right? Should be a flashback to Bio 210. In this case, that's very, very much true, okay? When we consider the product, the waste at this point, it is technically still chyme, okay? But a lot has been extracted from it already, right? Remember, in the stomach, water is already pulled out of the bolus. By the time that chyme emerges and passes through the three segments of the small intestine, more water is being absorbed out, as well as all of those monomer nutrients are being pulled out of that chyme. So if we pull all of those liquid components out of the chyme, by the time that chyme, the waste, reaches the large intestine, it's going to be much thicker and much drier in nature, okay? So this is where form meets function. Because it is thicker and drier waste that now has to be moved through the length, through the segments of the large intestine and excreted from the body as waste, we need help, okay? Peristalsis still needs to be effective. We still need to move this waste product, okay? So the large intestine is made up again of simple columnar epithelial cells that are going to also lay down, because of goblet cells being a part of simple columnar epithelial tissue, the goblet cells are going to lay down thick mucus, kind of put down a coating of the large intestine of oil, if you will. It's mucus, but it's a slippery surface. It keeps things slippery, eliminates friction so that thicker waste can move through the large intestine a little bit easier, okay? Secondly, let me talk first. The large intestine, unlike the small, does not have the circular folds, the villi, the microvilli, any enzymes coming into it. All digestion is completed for the most part by the time we get to the large intestine. Again, we're talking about waste for the vast majority of instances. with the exception of, like I mentioned previously, a little bit of water may still be absorbed from the large intestine, but even the bulk of the water has been absorbed from the stomach and the small intestine. So they're very different in the inner lining of the large intestine. All right. But related to what I was saying as far as form and function, remember we talked about the haustra, these very muscular individual bands that make up the large intestine and create a very, very different visual appearance of the large intestine compared to the small, right? Those thick muscular wall bands called haustra, combined with the mucus that's laid down by the simple columnar epithelial tissue, allow for the propulsion, peristalsis, to continue to be effective, moving that drier, thicker waste product through the colon and eventual ejection from the body. Okay? Form meets function. All right, so the stomach, if you remember, on average, had food reside within it around somewhere around 4 hours, depending. In the large intestine, that waste product is going to hang out significantly longer. You can see roughly 12 to 24 hours, depending again on different circumstances, okay? Again, there's no additional food breakdown that's going on except what bacteria will digest, okay? And those bacteria that are released from our appendix, those microbes that make their way, and millions of bacteria exist in our colon. Okay, they're going to continue, and you can see this is important for your exam to remember this, that those bacteria are going to continue to work on indigestible carbohydrates, okay? Things like heavy starches that so far along the alimentary canal haven't been able to be digested or thoroughly broken down, okay? So just know for the exam that in the large intestine, there's really no other nutrients being absorbed or digestion occurring other than bacteria continuing to digest those largely indigestible type of carbohydrates, okay? Real heavy starches and fiber that are real difficult to break down. And again, like I said, water is also absorbed, but to a much lesser degree than the other two places. Okay, I talked about previously the contractions of the haustra, that they're very, very important, again, to create peristalsis, to move that thicker, drier waste product through the large intestine. Okay, we also have what is called in the large intestine the gastrocolic reflex, okay? And this is that same intrinsic gut brain, the intrinsic nervous system that I talked about a couple of times now. Gastro meaning stomach in this case, colic meaning colon. Okay, this is that connection, that gut brain, the nervous system within the digestive system where one part can talk to another. And it's the exact same example that I've used for you guys previously. When the stomach takes in a large amount of food, the sensory receptors that are built into the walls of the stomach will use that enteric intrinsic nervous system, the gut brain, and send a signal to the large intestine to speed up contractions and peristalsis within the colon to clear itself out, because there's a massive amount of food in the stomach that's going to need to be making its way toward it, okay? This is exactly what we've talked about multiple times before. The term is new, gastrocolic reflex. All right, and the last of your two quiz questions for today. So, which of the following is the role played by bacteria that exist in the large intestine? All right. So, guys, that wraps up the digestive system. Okay. Next week, Tuesday and Thursday, I'm going to emphasize this one more time to you. Don't miss Tuesday and Thursday of next week. metabolism. We're going to talk more about further chemical breakdown of all the macronutrients. We're going to talk about the conversion of one nutrient macromolecule to another and all the processes, what they're called and how it occurs. We're going to talk about the reassembly of monomers into macromolecules now that they're in the bloodstream. And we're going to talk about, very importantly, the use of macromolecules, nutrients, and the conversion of those nutrients to ATP, which is the fuel that drives all of our cells of our body. There are several, if you didn't hear it and have it explained to you, it could be complicated, but it starts with things like the citric acid cycle, the Krebs cycle, into the electron transport chain and oxidative phosphorylation, some new terms, new processes to be sure, glycolysis, glucogenesis, gluconeogenesis. You're not going to want to miss those lectures if you expect to do well on exam number 3. So I just really want to warn you, so no one can say, wow, I was really surprised. Don't be surprised. Heavy lifting next week. Okay, so guys, are there any additional questions or any questions, period? All right, well, outstanding. Guys, enjoy this beautiful weekend. I understand tomorrow we're supposed to get up into the upper 70s, low 80s. By Saturday, we're supposed to get into maybe the upper 80s, they're saying. Crazy. Only South Carolina in the spring, right? But enjoy this beautiful weekend. Have a safe weekend, an enjoyable weekend. Send me your extra credit points so I can give you those extra credit points for exam number 3 within 15 minutes. And we will see you guys all on Tuesday. Don't forget to study for your lab quiz on Tuesday, as well as lecture. See you then.